Hi, Matt. Thanks for joining me again. I really appreciate it.
Absolutely. Thanks for having me.
What are N-glycans and how do they differ from other types of carbohydrates found in foods or biological systems?
Yeah, that's a great question. Glycomics is a interesting area of research right now. It's one of the things that most people think of when they think of proteins and even cellular proteins, dietary proteins is, maybe some post-translational modifications. The common ones people tend to think of are things like phosphorylation. Protein gets phosphorylated, turns something on, protein does set action. But the other interesting post-translation modification is glycosylation. It's the addition of this carbohydrate structure. There's two types. There's N-glycans and O-glycans. In our lab, we focus on the N-glycans, and I'll tell you a little bit why later on. But these N-glycans are very structurally diverse carbohydrates, very big carbohydrates. They affect things like the protein stability, the way it migrates through the cell, the way it communicates with other proteins, and so forth. You tend to find a lot of them on surface or membrane bound proteins that they'll get glycosylated and they kind of signal to either other cells or immune cells and all that good stuff of where the protein is and so forth. But what's interesting about these is they look like fibers. They resemble fibers in the sense that, when we eat, let's say, actually, it's funny, I'm having a protein shake right now. And as I'm drinking this whey protein, which is derived from milk, these proteins have these N-glycans attached to them and they resemble fibers. Now, what's different about these carbohydrates, because they're these complex carbohydrates, probably anywhere from about, let's say, 7 monomers, 7 sugar units, up to maybe like 15. They could get pretty big. And the possibility of constructions are practically endless because you get, when people think of sugars, they think of maybe glucose, galactose, fructose, just these three, but there's a bunch of sugars, sialic acid, galactose, and acetylglucosamines. There's all these derivative sugars. So they could be constructed in these very cool ways. And the unique thing about these N-glycans is that they're bound to protein. Unlike other sugars in my protein drink right now are free floating. So you got lactose in it, maybe some other sugars, but they're not bound to any protein. That's what kind of makes these N-glycans rather unique, especially from a dietary perspective. If I were to ingest this, the question is, okay, well, does it get absorbed? Well, no, because it's bound to this amino acid. It's too big. Our body doesn't have the enzymes to break down this big sugar. Therefore, it acts as a fiber, makes its way to the colon. And that's where they're kind of unique in that sense. Very long explanation, but
No, that's great. I appreciate it. What motivated you to study N-glycans and common dietary protein isolates? And I know whey is one of those examples, as you just mentioned, but there's also egg white, soy, and pea protein as well.
interestingly enough, actually, before I started my PhD, I super into the gym. One of the things in high school, I played football. So I was like, okay, I got to get bigger. I got to make sure I'm putting on muscle so I could go to the league, right? That's what everybody thinks. And then one of the things I wanted to do when I started my PhD was I actually wanted to come out with a supplement company, like a supplement line. I want to research into these dietary proteins. What's the best protein? What amino acids do we need to build the most muscle? All this good stuff. And then I had the opportunity to work with my advisor now, Dr. Stephen Freeze. As interesting as it was, he's a gut microbiome researcher, but he was working with whey protein and milk proteins. And I was like, okay, this might be a really good opportunity. And that's how I kind of got in there. And what was funny about that was I had no idea about glycomics or in glycans. I just wanted the protein side of things. And that kind of kicked it off. And then, as I got interested in the glycomic side of things, I realized that the constructions, the structures, it's endless. It's such a hot new field. They're looking at, for example, this is another cool one, although it's not dietary proteins. They're looking at glycosylation patterns in cancer, certain glycans on cell surfaces. The glycans on them tend to change. And this is a cancer cell's way of manipulating the immune system to pretty much say, hey, we're fine. No, nothing's happening here. So it's interesting. It's cool because then you start to see these things and you could connect the dots. Okay, what's going on? Why is this happening? How do we stop it from happening and so forth?
Yeah. protein's done a lot of great marketing for itself
Oh yeah.
recently in terms of everyone paying attention to it. For some content I've come across that in terms of protein shakes like you're having, that seems to be a very high quality source of protein where some people might think, oh, like regular natural foods are better. But in terms of how it's absorbed and processed in the body, it sounds like some of the whey protein isolates are about as high quality as it can get.
Yeah, I would agree with that, especially in nutrition. One of the interesting things is protein's so complex in the sense that, okay, how fast does it get absorbed? How quickly does it spike muscle protein synthesis? And you could get lost in a whole area there too. So that's really cool stuff.
I was hoping you could walk our listeners through the main steps in your experimental work through just to give them a all around picture of the work you've been doing.
Yeah. So, again, unlike free floating oligosaccharides or sugars, you can isolate those rather easily, but there's one extra step with N-glycans, and that's because they're bound to that protein. So the first thing we tend to do, depending on the protein source, is, even in protein powders, right? Even in protein shakes, you'll notice that it's not pure protein. There's always some sugar in there to make it taste good, maybe a little bit of fat. So the first thing is we have to purify that protein out. And the way we do it is we try to start with a source that has very little sugar. And then we'll do ethanol washes, and ice cold ethanol usually takes the free floating sugars out into the ethanol solution. We precipitate the protein and we do a few of those washes. And then we confirm with what's known as a phenol sulfuric assay that there's no more sugar in that protein other than the N-glycans. And phenol sulfuric reactions, they release a colorimetric assay when there's sugar in the system. So that's how we check to make sure we got all the sugar out of the ethanol phase. Now we have just pure protein with N-glycans. So from there, what we do is we have to use this enzyme, rather expensive, rather cool. And we actually just published some cool work on using gut derived enzymes to do the same thing. But it's known as PNGase F, and it's a glycosidase. What it does is you have this cute little N-glycan structure coming out of the protein. It'll cut it off just like a pair of scissors and remove the whole N-glycan fully attached. So it doesn't chop up the sugars. It actually just removes that whole sugar from the protein. From there, we purify the N-glycans. And now when I say I mean my collaborators over in Turkey, Dr. Sarjan Turav and Dr. Mehmet, really brilliant people. So what they do, they take the N-glycans, they purify them, and then they run it through. And we've used both, actually. We have some published work using MALDI mass spec. And then we have some the work that we're kind of discussing today using QTOF, HILIC HPLC. So there's a lot of ways, all trade-offs. There's no perfect solution, but there's these cool trade-offs. So then from there, we actually label the N-glycans with some sort of tag. We've used 2 AA tags and we use procanamide tags. And then from there, once they're purified and they're tagged with a little tag, we could run that through the HPLC or the MALDI and then use the mass spec to identify glycans. And it's semi-quantitative. So it'll tell us about how much through the intensity, but it doesn't give us a perfect, this is exactly how much you have per protein.
Are there some key challenges or interesting troubleshooting experiences you've encountered in this work, Bo? And this could be on your side or maybe what your collaborators in Turkey have reported on.
Yeah, that's a really good question. I would say that our collaborators are really, really good at doing this. So they have it down rather well. The biggest trouble is when you look at protein, glycans in the gut. Mucin protein is a beautiful example, but only a very exclusive example where 85% of mucin protein in the gut is carbohydrate of glycans. So you get a lot there per weight of mucin protein. But that's not quite the case with dietary protein. With things like lactoferrin, which is in whey protein, things like colostrum in whey protein, you could get about 1 to 2% by weight carbohydrate of those N-glycans per protein. So huge difference. So one of the hard parts for us, especially when we want to study, okay, what are these N-glycans doing in the gut when they're removed from the protein? The yield. How can we get a lot of N-glycan without having to take a pound of protein? And then how do we purify it without having some of the nice equipment that they have over there? So those are the big challenges. We actually tried doing that in the lab with PNGase F. We used another enzyme that we actually derived from the gut that we found using metagenomic sequences. And the yield is just a bit tough. We're still kind of working out the kinks there, but that's the biggest challenge. And from a practical standpoint, I think purifying the proteins, it sounds easy, some ethanol washes, but you got to be it's tedious. You don't want to introduce any sugar from anywhere, and it gets a bit tedious. Other than that, it's the yield on these carbohydrates. How can we get a lot of them separated from the protein and purified so that we can play around with it in vitro gut microbiome samples?
Earlier, we touched on the different and glycan profiles. What are the main differences between the animal derived, whether it's whey or egg white, versus plant derived, whether that's soy or pea protein?
Yeah, that's a great question. So what you tend to notice in what I like to call like simpler organisms, plants, things like that, is they don't require as many sugars to construct their end glycans. And what I mean by that is more the diversity. So what we found when we sequenced or when we used mass spec on the pea and the soy protein is it was mainly mannose sugar that constructed those end glycans. Now, granted, it was different configurations. They had different structures, but it was almost exclusively what's known as high mannose or oligomannose end glycans. Now, when you look at the animal derived, these higher order systems with animals, egg white as well, you could probably see it in some insects. Insects are kind of interesting too. But in the animals, what you tend to find are, again, bigger structures, but way more monosaccharide diversity. Sialic acid, you have galactose and acetylglucosamines and acetylgalactosamines. So you get these way more diverse monosaccharide configurations, which makes it a bit more interesting when you think about it in the context of, okay, well, what effect might that have on the gut microbiome? We actually have another paper on that where we compared the synthetic whey protein that's produced in microorganisms compared to the natural traditional whey protein. And what's cool about that is we fixed the confounding variable in protein because the sequences were about the same. They were both whey protein. However, since one was produced in microorganisms, those post-translational modifications only used high mannose and glycan configurations. So we had a nice set point where we had whey protein with high mannose and glycans, and then the traditional whey protein with very complex and glycans. And we noticed that there was a pretty big difference on the gut microbiome, which is cool. The more diverse structures tend to have higher alpha diversity on the gut microbiome than the lower diverse structures, which, kind of makes sense within itself.
How might your findings about end glycan diversity and structure influence future research or applications in areas such as nutrition science or food in general, microbiome targeted therapeutic?
Well, the cool thing about these end glycans, especially understanding their structure and diversity, is we could start to disentangle which structures certain microbes prefer, and we could start to kind of look at microbiome-based therapeutics in the sense of what's known as these new cool symbiotics, where certain microbes prefer certain prebiotic structures that induce the most activity in the gut. And we could start to begin to pair these microbes with certain structures, and we could disentangle these complex relationships in the gut to really maximize the metabolic output of the gut microbiome. That's kind of the goal. We want a lot of these short chain fatty acids and beneficial metabolites, but by understanding the diversity of these structures and understanding the structures themselves, we could start to pair them with certain microbes to help maximize that metabolic activity in the gut.
I know you've been at this work for a while now. What laboratory skills or maybe theoretical knowledge from your undergraduate studies have really helped you and been most relevant, let's say, in your research in glycomics and biomolecular analysis in general?
Yeah, there was two classes that really stood out in terms of starting to understand glycomics. It's such a nuanced area. There wasn't a class on glycomics, and I think we're going toward that way. But there was two classes that really helped. One was biomolecular physics, and the other was biomolecular engineering. And both of these things really kind of hit the root of post-translational modifications, specifically glycosylation in my case, to start to understand, okay, what are these attachments, these protein bound glycans, what are commonalities between them, and why do they get constructed the way they do? And then from there, we could kind of work our way further in the type of work that I'm doing.
The main audience of this podcast is undergraduate students who are analytical chemists. Do you have any advice for those students who are interested in careers in biomolecular chemistry, or maybe even people who are just generally interested in doing chromatography and mass spec in their future work?
Yes, I definitely think that the first thing is understanding what both sides of the equipment does, right? What does mass spec do in terms of trying to understand what compound you might have, or using a HPLC or a GC? What exactly is this tool doing to try to elucidate whatever structure I have, and understanding the trade-offs? Okay, what should I use, a MALDI TOF, or should I use a QTOF, or should I use a HPLC? All these different ways to try to understand, okay, what's best for what I'm specifically looking at.
Are there any common misconceptions or maybe just general lessons learned about and glycan analysis that you think the audience should keep in mind?
Yeah, that's a really good question. So we have, we currently out of the lab have two publications looking at N glycans from the same dietary protein sources. And one's using a MALDI-TOF time of flight and then paired with a mass spec. The other's using a HILIC HPLC QTOF. So we're looking at the same proteins and the same N glycans. However, there's trade-offs of both equipment. There's certain MALDI-TOF is really good for sensitive quantification. We elucidated a lot more structures with the MALDI-TOF. However, the QTOF was a lot more, I guess, consistent in what compounds or what N glycans that they found. So again, I think it kind of comes at the root of understanding the trade-offs between equipments. Okay, well, what's going to be the best for my type of analysis, and how could I use this for what I'm looking at?
I think you mentioned that earlier, and I thought it was a really great point that there's not going to be, in a lot of cases, a perfect fit in terms of the analyzer you're using versus the answers you're trying to derive. I think that's something that's not always talked about. And it's probably because of how I'm approaching it I'm asking a lot of academics about their research process. And it's easy to say why using HPLC, for example, was the best tool, but that's not to say you couldn't have gotten helpful answers using another type of analyzer. Does that make sense?
100%. Yeah. Yeah, I totally agree with that. Especially with, like if there was a perfect solution, I think, everybody would be singing and dancing and happy. But yeah, it's always a matter of trade-offs. Okay, what's the best piece of equipment to answer my question the best, So
Matt, I really appreciate you taking the time out of your vacation in sunny Florida to speak with me and update the audience on your work. And I hope to keep up with you and learn about what you're up to next year.
Yeah, thanks, David. Very happy that you worked with me all the time. It was great. And I'm glad we finally got to do this and catch up and chat a little bit.